Yeah, sure. Well, there's a specific way that you need to design an experiment in order to show that there's a virus that exists. And it's been done several times in lower organisms, like in bacteria or algae, where they've found what they call viruses. Interestingly, those are not actually thought to cause harm to those organisms, but may actually be a way to help their survival when they're in a difficult environment, like under a lot of stress. But through those experiments, you can see that you can basically take the source of the virus, which in that case would be a mixture of those bacteria cells and the bacteriophage particles, which are what they call a virus in bacteria, and they filter it. And filtering is a very powerful way to purify the virus particles because they're so much smaller than the cells. So, for example, they say that a virus particle is on the order of 100 nanometers, and that's a billionth of a meter is 1 nanometer. So very, very tiny. The cells are much, much larger. So by putting them through a filter, which just has tiny pore sizes that only small things can fit through, you can separate it from the rest of the cells. And then once you have it purified, you can then characterize it. In other words, look at what the chemical composition or the structure of it is, and then you can extract any genetic material and characterize that. When they've done the experiments claiming to isolate the SARS-CoV-2 virus, which is the name of the virus that they say is associated with COVID-19, the disease, they don't follow that procedure at all. They, in fact, don't purify any virus particle during the entire experimental procedure. And this is a fundamental principle of science that if you want to measure a variable, you have to separate that variable from the other ones, right? So you design experiments specifically where you separate that one variable. There's another principle of research that they don't do called a control. So you always have a control so that you know that whatever you're measuring is not present in the control. So it's like a negative sample. So in order to show that there's a virus particle in these experiments, what they do is they take the fluid from the patient, which has a mixture of a bunch of things, right? It's just lung fluid. So it's got, sputum, it's got different kinds of cells, it's got genetic material like bacteria cells, human lung cells, immune cells. And they mix it with a foreign cell culture, usually, like monkey kidney cells. And they incubate that and grow it in culture. And they also add antibiotics to that. And then after they grow it for several days or a week or more, they look at it under the microscope and they see little particles. But the thing is, we don't know the source of those particles at all. And we know that when you mix cells in a culture with antibiotics, that they make exosomes. And exosomes, I can explain what they are in more detail, but they basically look the same as virus particles are alleged to look. So when they look under the microscope, how do they know if they're seeing the exosomes, which are going to be there, or something else, which they're calling a virus? So if they just did a simple control experiment where they had the same cells with the antibiotics, but didn't add the lung fluid to it, that would be the control experiment. And then they could show pictures of that and show the particles there and see how they look different from what they are calling the virus particles. But they've never done that. And I suspect the reason is because they would show the same exact particles in that experiment, and then they would say, oh, well, we didn't find a virus. And that's not what they want to say, because the way you get famous as a scientist and the way you get more grants is that you write a paper which says you isolated a virus.
And that's very interesting. I'm not familiar. I'm not sure if you're familiar with John Rappaport's work. He's been writing about a lot of this stuff, and he said something in another vein of thought, and the idea that they're not doing what they're supposed to, that they'll ultimately be able to show to some degree that there is a certain amount of it, but not be able to tell whether or not there's enough of it to suggest that someone is infected, as well as the discussion that many people have mentioned Koch's postulates and the same idea that they're not fulfilling that part of the scientific, standards ultimately. So there's a lot of things we can see here that they're just not doing the way that they're supposed to, which I think is very suspicious to start off in general. But before I get to some other questions, so on what you're saying, which is really fascinating, are you saying that in a general sense that this is not a virus, or that there is that viruses don't exist? Because I'm really interested in that, because I see a lot of really educated, well-researched people out there suggesting that we just don't understand what viruses are in general. So is that where that is essentially?
Yeah, well, I think one is important to separate out what is the proof of the existence of the virus versus how they do the testing. Because what you were mentioning about John Rappaport's comment has to do with the testing. And that assumes that they've already have a virus. And then they can, say, well, how much of it is in your body? Is it enough that to tell us that there's active disease or not? But that's not really very important if there's never been a virus proven to exist in the first place. So I've looked back to many papers of prior coronaviruses, and I've looked back at many other papers claiming to isolate other viruses. And it's important to mention that Koch's postulates, right, which was put forth by the germ theory scientists, so basically the same scientists that are doing this research, they came up with the rules. And what they found was that Koch's postulates existed before they ever saw something that they called a virus under the microscope, because electron microscopes weren't invented till the late 20s, and then it was until the 30s that they saw these particles that they called viruses. And what they realized is that they were unable to satisfy Koch's postulates for any virus. So this prominent virologist named Thomas Rivers wrote a paper in 1937 basically saying, we can't really do this successfully, so here's a shortcut, and made the criteria easier, and that's sometimes called the Rivers criteria. And in a paper in 2003 for the SARS-CoV-1 virus, which is supposedly related to the current virus as an ancestor of sorts, and they say that that was responsible for the SARS outbreak in 2003, which I think only killed 800 people. They claimed that they satisfied Koch's postulates in that paper in 2003 for the SARS-CoV-1 virus. And then it's funny because they used the word Koch's postulates in the title, but then right in the article, they said that they used Rivers criteria, which is not the same. And they also misinterpreted Rivers criteria, saying that it had 6 components when it actually didn't have that many. And then they claimed to prove it by referencing other articles and saying that they did experiments themselves to prove 4, 5, and 6 criteria. But when I scrutinized that, I found that actually they didn't really prove 1 criteria definitively out of the 6. And the most important one is been universally proposed in every rule that I've seen, which is that you have to isolate the virus from the sick person. And I've not seen that be done for any virus that I've looked at. And I've been in touch with several other researchers who have been looking at this. I mean, many others, including some virologists and other MDs. And they also have not found any evidence for any virus being proven to exist. And even the German Supreme Court ruled on this, because one of those virologists, Dr. Stefan Lanke from Germany, he put out a challenge that if anyone can prove that the measles virus exists, that he would pay $100,000. And several people challenged him, but of course they didn't meet his criteria, which were not impossible to meet. They were very reasonable. So what happened is someone sued him because they thought they met the criteria and didn't believe his rejection. And it actually went up to the German Supreme Court. And so the German Supreme Court had to get experts from the Robert Koch Institute, which is quite ironic because that's like their CDC in Germany. And it's named after Robert Koch, who is given credit for putting these rules out in the first place. So they had to have scientists from there basically testify in the court that they didn't satisfy the rules of proving the existence of measles. So the German Supreme Court ruled that measles does not exist.